Hello everyone. First, I would like to thank everyone who joined us today for the seminar. We are pleased to have with us today Professor Rebecca Wingert. Professor Rebecca, thank you so much for being here with us and accepting our invitation. And just a brief introduction before you start, Professor Wingert has a PhD in cell and developmental biology by the Harvard University, and she was a postdoctoral fellow at Harvard Medical School at the Massachusetts General Hospital. And since 2016, she's an associate professor at the Department of Biological Sciences at the University of Notre Dame. So at this institution, she's also the director of the graduate studies and is part of the Association for Women in Science. In her research, she studies the genetic and molecular mechanisms that control how renal stem cells accomplish kidney formation, impact the kidney homeostasis, and facilitate kidney regeneration following organ injury. So to study this, her group used the Zebrafish model due to their many similarities with more complex vertebrates like mammals, and considering a high degree of conservation between gene function and basic cellular processes between zebrafish and mammals. Her group research centers around the following major areas: kidney development research, kidney regeneration and aging research, and stem cells, renal birth defects, and heart disease. So Professor Rebecca, thank you so much again for being with us today. For those who are watching us, we will be open to questions at the end of Professor Rebecca's talk. So you can put your questions in the meantime at the chat here or in the YouTube platform. So thank you so much, Professor Rebecca. You may start your presentation.
Lisa, thank you for the wonderful introduction. That was very, very kind. And I'm so pleased to be visiting you all today and to have a chance to tell you about some of our research. The work that I will be telling you about today is unpublished work. And so we're really excited to we're in the process of trying to publish it, and we're excited to get feedback on it. So I welcome any questions and feedback that you might have. So today I'll be talking about using zebrafish to study the genetics behind how cells in our body form structures called cilia, and how they decide what kind of ciliated cell to actually make. And the view you're looking at here is a beautiful confocal picture that was taken by one of my graduate students, Hannah Wesselman. And she's the student who did the majority of the work that I'll be presenting today. And I'll show you her picture at the end in the acknowledgements. But this is one of her very beautiful images of the zebrafish kidney, where she's done stains so that she can see the cilia that are actually projecting off the different cells in the tube of the kidney, which is running here across the slide. So to get started, what I'd like to do is just remind everyone what cilia are and tell you a couple key bits about what their functions are in the body. So here on the left, you're seeing a drawing of a cilia structure. These are thread-like projections. Sometimes they're referred to as antenna-like projections that emerge from the surface of the cell. They're found on the apical surface of the cell, and on most vertebrate cells, they actually have this kind of organelle. And cilia themselves are made up of an axoneme, which is at the center of that thread-like projection, and it has an anchor to it so that it has a way to structurally be organized, and that is known as the basal body. So you can see that term basal body here. And then the axoneme that projects off of that basal body is comprised of microtubules. And these structures are built by what's known as interflagellar transport. So those are some of the other proteins and these components here, which are moving up and down the cilia. This is part of the trafficking that happens to actually build cilia and maintain it, and also is involved in ciliary function. So these structures are architecturally quite complex, with many hundreds of proteins that make them up. And these cilia are very important in our body, and they're important in a lot of different places in the body. So ciliated cells across the body have many essential roles. And on the right here, this diagram that I'm showing you is actually a map of where cells are located that have multiple cilia. So sometimes a cell will have one cilia that's projecting off. Other times there will be bundles of many of these cilia. So there might be a bundle of 10, there might be dozens, in fact, of these cilia that are projecting off of the cells. And so in places across the body that are shown here, again, these are places where there are multiple cilia. And these multiciliated cells, or I'll talk about them and use the abbreviation MCC, they're super important in structures such as the brain. So in these places, or in that location, these multiciliated cells, the cilia actually move. And the beating of the cilia, the movement, will move fluid and circulate the cerebrospinal fluid. And this is essential for brain and spinal cord function and health. Similarly, multiciliated cells are located in the respiratory tract. Here they move mucus, and this is part of a way to clear debris and different pathogens and such that actually move into our lungs and respiratory tract. And so you can see that there are those bundles, they interact with the mucus layer to actually help facilitate the movement of that mucus. Multiciliated cells are also found lining fallopian tubes. And so in that location, they actually usher along the egg as it moves from the ovary to the uterus. And so again, these are all places where multiciliated cells are located. Monociliated are located, again, all across the body as well, where they have key functions. Really, the functions of cilia will bin into categories such as sensory functions. So based on how that cilium is influenced by its environment, it can be a mechanosensor. So a lot of monociliated cells, that cilia is a sensor for the cell to figure out and get information about what's happening in the extracellular space. And in other cases, that single cilia might be motile and move itself, or in the case of these multiciliated cells, they all have cilia that are moving. And so collecting information, that signal transduction that would happen afterwards is one important function of cilia, and the other major function is movement of liquids and other components of the body. So the key point that I want to emphasize is that cilia are very, very important for organ function, and they're also very important for development. And what's not known fully is all the components that go into making cilia. So that process of how you form a cilia, how a cell knows when and how to make it, and has all the right instructions in the right sequence to form cilia, to make them stable, etc. That is not fully understood. So despite many tremendous advances in the last couple of decades, there are many gaps in knowledge that remain. And so my lab came across this topic and question as we were studying our organ of interest, the kidney, because the kidney cells are ciliated, and we've been utilizing the zebrafish as a platform to learn about these fundamental mechanisms of how cilia get made. Okay, so the next thing that I want to provide as background is just to tell you a little bit about the zebrafish. So this is an animal model that is just a wonderful system to study many questions in development, as well as to study and model human diseases. This is because zebrafish, so here I just have some drawings of zebrafish. You'll see some embryos in just a few minutes. But these embryos are amazing for many reasons. Fundamentally, zebrafish have very high genetic conservation with humans. And so this means that genes that we study and seek to understand their functional roles in the zebrafish, a lot of that information is recapitulated and is applicable to understanding human biology. So they've got high genetic conservation with humans, but in terms of an animal model, they're extremely tractable for experimental work because these embryos are born outside of the parent. So the male and female zebrafish will actually spawn their gametes into the water. And so this means a researcher can collect the embryo when it's first fertilized and observe development from the very beginning. In addition, these embryos develop very quickly. So I'll draw your attention here on these drawings to the ages that they are depicting or representing here. So already at 12 hours old, so 12 hours from the time of conception, this animal has developed such that it has a very obvious head structure and a trunk that's actually wrapping around the central yolk ball. So this darker brown in the middle is representing a large ball of yolk, and other molecules, of course, are in there that support development. So at 12 hours, the embryo has this appearance here on the left. By 24 hours, there's tremendous growth and organ development. And you can see even more so a very discernible head and eye and a long trunk, and organs such as the kidney are made by this time. So it's a fantastic model because these embryos develop outside the parent and they're very rapid. They're also quite large as embryos go. And so this means that they're very readily observed with microscopes in the lab and can be worked with because of their size, worked with very easily. Okay, so at 12 hours old, the zebrafish has been used to study cilia development in this organ known as the Kupfer's vesicle. So that's the inset that I'm showing you here. This is a picture that Hannah took of ciliated cells, monociliated cells that sit in this vesicle. This is also known as the node, and this is important for setting up the body plan of the embryo. So in this location, there's actually fluid that the cilia are moving, and they're moving molecules that help the embryo to discern and set up the left-right axis. At that 24-hour time point, shown here on the upper right, that kidney, as I said, is formed, and I'll show you some other schematics to help you visualize the kidney in a second. But within that structure, we have ciliated cells, which are formed. There's two populations, both mono and multiciliated cells. And so by 24 hours, we can see them within the kidney organ. And other labs have looked and utilized zebrafish at later days of development as well. So just as an example, at the 4-day time point, people study and use the zebrafish ear, known as the otic vesicle, and different structures in that ear have cilia. And there are also sensory structures that exist along the length of the zebrafish embryo, known as the lateral line, comprised of these structures called neuromass, which are also ciliated. So zebrafish provide a couple very expedient organs and settings in which to look at cilia development from earlier stages of less than 1 day over the course of the first few days of development. So with that background to cilia and to kidney and where cilia are, in some places people have studied in the zebrafish, I'm going to jump right into the unpublished data that I'll share with you today. And I think I neglected to put a link up, but I'm happy to share that afterwards. So the story that I'm going to tell you is about this gene known as estrogen related receptor gamma A, so it's abbreviated ESRR gamma A, and this is work that we have posted on bioarchives, and we're in the process of sending it to journals and having it reviewed and get feedback on the work. So you can go to that bioarchive site, and like I said, I can provide that link at the end for any who are interested if you'd like to learn more, because I'm going to give you some key highlights here, but not every single thing that Hannah has done. Okay, so I'm going to talk about this estrogen-related receptor gamma A, and why did we select this particular gene and its protein product as a topic of study. It actually came because Hannah was interested in studying estrogen signaling in the kidney, and we started looking at the estrogen receptors and studying those, and noticed this related gene, and got very curious about its function because of a couple of key things about this gene that were actually published already. So although this is a estrogen related receptor in the name, this particular gene encodes an orphan nuclear receptor. So it's the ligand that activates this receptor is not presently known. But a couple of things caught our attention about this factor. The first is that it was expressed when we looked at our databases in the zebrafish kidney. And so we could see expression in these embryos that I'll be pointing out. The expression of the estrogen-related receptor gamma is in purple. And we could see expression of RNA transcripts here with this whole mount in situ hybridization assay. We could see that in the kidney precursors at many stages of development. Here I'm just showing 2 stages. This is around 20 hours and this is around 24 hours, a little bit later of development. And in each case, we saw that cells within the kidney were expressing this transcript. And this was paralleled with the human embryo. So data from a resource known as GoodMap. This is a group of labs and researchers who have been curating information about genes related to genitourinary tract development. And so we looked in that online repository, and lo and behold, in the human kidney at about week 17, there was very high expression of this gene in the precursors that make functional units in the kidney. So these tubular precursors, which are going to make the kidney, had very high expression of estrogen-related receptor gamma A. And there was actually a published study that we then found and looked at in the literature, where a lab reported that if you knocked this gene out in the kidney, this was related to lethality of the animal. So actually, if you broadly knock out the gene, this causes embryonic lethality. But if you specifically knock it out in the kidney, you get these enlarged kidneys because the kidney is filled abnormally with fluid. They have this cyst accumulation. So from our from these pieces of information, we were really, really curious about this gene and its function in development. So that got us interested in the kidney, and you'll see that we found a link to cilia. And so that's where we're going with this. So what Hannah and I decided to do was to utilize the zebrafish embryo as a way to interrogate and identify what the functions of this gene are in kidney development, to get a better idea about what happens in the kidney. And so Hannah designed a couple of different methodologies and tools to interfere with the normal expression and function of this gene in the zebrafish. And here on the right, what I'll show you is that she's designed several different tools. So this gene is encoded by 3 exons in total, and Hannah designed some tools that would interfere with production of the protein using morpholinos. So in this red color here, these are the locations of her morpholino tools, their targets. And she's also designed tools to edit the gene using CRISPR-Cas9, and designed ways to interfere with exon 1 and introduce premature stop codons within that sequence. So the data that I'll show you today are pictures primarily of animals that we would refer to as morphans, where she did transient gene knockdowns of estrogen-related receptor gamma A, but she's identified identical phenotypes in her CRISPR models. But just for the sake of simplicity, I'll just show you examples of animals deficient in this gene, but they were generated by morpholino knockdown. Okay, so when Hannah interferes with the expression of estrogen-related receptor gamma A, she saw a couple things that jumped out at her right away. And those were that these animals developed unusual accumulations of fluid in the body. In this embryo, you can see that there's actually fluid that's denoted here by Hannah with this asterisk that's around the heart. So this is a pericardial accumulation of fluid, or a pericardial edema. And anytime we see a fluid imbalance, this is a potential sign, a potential harbinger that there is a defect in kidney function in the animal. But she also saw other kinds of defects in the animal. And the one that I'll point out is in the otic vesicle structure. And so she's has enlarged pictures here, or pictures at a higher magnification, where you can see that compared to the wild type embryo, the otic vesicle has an abnormal structure to it. And this type of phenotype, where the otic vesicle is disrupted and these different components are fused, this is actually a sign that that is suggestive that cilia are not being made correctly in these animals. And because of the fluid imbalance in the animals, we hypothesized that perhaps the kidney was also affected, and that cilia, which would be the common thing between the ear and this fluid around the heart, that cilia could be at the root of these different phenotypes that were occurring in the animal. Okay, so we took then a closer look at the kidney, and on this next slide, just to orient you, Hannah utilized a one-day-old animal and fixed the samples, and then took sections through the kidney so that she could look at the cells in the nephrons, in the nephron functional units of the kidney. And so in the kidney, and you'll see this more in a few moments, the nephrons are comprised of two major different cell types. And those are cells that have just a single cilium, that monociliated cell with one cilia projecting off the surface, as well as multiciliated cells, which have multiple cilia projecting off. So first, Hannah took a cryosection, and actually took cryosections so that she could look at the cross section of the nephron tubules. And when you look at the cross section, there are typically 5 cells that make up the sides of the kidney units. And what she observed in her samples was that in the wild type kidneys, these cells had very beautiful cilia that were projecting off of the apical surface into the lumen of the nephron. So the lumen here would be in the very center. So these cilia projecting very nicely into that lumen. But in animals that had the knockdown of estrogen-related receptor gamma A, she saw a decreased amount, what appeared to be a decreased sort of amount of ciliated structures here. So less fluorescence that corresponds to the acylated alpha tubulin that lets us visualize the axonemes of the cilia. And when she combined that with stains for gamma tubulin, which is present in the basal body, what you could see in the cell, what she could see in the cells of the deficient embryos was that there were basal bodies on cells, but some of these appeared naked. They did not have a cilia associated with it. And so these observations led us further to hypothesize that cilia formation was being affected in the kidney, but it sort of seemed like the basal bodies were just fine. We needed to go do some more analysis, of course, which I'll show you. But in other stains that I won't show you here, these cells have very clear apical and basal sides. So this is not a reflection of the cells being at a stage where they haven't set up their polarity yet. They have very clear apical and basolateral distribution of other proteins, but cilia formation is disrupted. Okay, so to look in more detail at the cilia, and actually to be able to really measure cilia length and quantify the numbers of basal bodies and whether they were ciliated or not. What Hannah did was look in whole mount, because there she, with the confocal, can actually image through the entire nephron and then zoom through the Z-stacks to actually reconstruct cilia and identify the locations of basal bodies. So when she looks in whole mount, what she can do is look at more regions of the nephron in the same animal. So instead of with the cryo sections, you only get small glimpses of what's happening at these different locations. With the whole imaging, she can survey the broad landscape of cells in the kidney. And the key thing that I want to point out to you is that within the zebrafish kidney, there are regions where multiciliated cells exist, and other regions where only cells with a single cilia exist. So the regions where there are multiciliated cells, that sits in about the middle of the nephron. And they're shown here with these purple colors again, and they're intermixed with monociliated cells. And so we refer to this as the proximal region of the kidney. And in this portion that is located at the end of the nephrons, we refer to this as the distal region. And in that region, there are only cells which have a single cilium. So when Hannah looked in embryos at with immunohistochemistry to look at cilia with alpha tubulin and gamma tubulin, she observed the following. So in her samples in the proximal, in a wild type, there are these beautiful projections of cilia into the lumen. And so in each of these images, what Hannah has done is with the white dots, she's outlined where the nephron is actually sitting, because the channels shown here, we just have the nuclei of the cells in the blue. So we're seeing the lumen here that's very full of cilia in both the proximal and distal regions of a wild type embryo. But when she knocked down estrogen-related receptor gamma, there were many fewer cilia here, and they were actually shorter. And she quantified that that there was an increased number of basal bodies that did not have any cilia associated with it. And so there's a significant decrease in the amount of cilia in both of these locations. And in part of her work to validate the tools that she has been using, she performed rescue studies. And so provided, in this particular case, did an experiment where she provided the normal transcripts that encode estrogen-related receptor gamma A to these animals. And when those normal transcripts are delivered along with the tool to disrupt the endogenous expression, you can see that she had robust recovery here, a robust rescue of the amount of cilia. So she could significantly rescue cilia length, and these that number of cells that had the naked basal body was returned to normal. And this was true in both the proximal and the distal. And so just for the sake of simplicity, I don't have all those quantifications shown here in the slide deck. But again, you can find them all online if you're curious about that or about how we do that. But these were significant changes that we could convince ourselves were specific to the tools that she's been using. So the key takeaway here is that in regions where you have both mono and multiciliated cells, there were ciliogenesis defects. And in this region where there were just monociliated cells, there are cilia defects in the absence of estrogen-related receptor gamma A function. So because of this region where we had this group of both multiciliated and monociliated cells, what Hannah also did was to explore whether the multiciliated cells were present or not. And to do this, part of her approach was to look by whole mount in situ hybridization at unique markers, so unique genes that are expressed in the multiciliated cells. And when she knocks down estrogen-related receptor gamma A, there's a significant decrease in the number of those multiciliated cells. You see many fewer cells here in the proximal region of the nephron, and they are rescued by the restoration of normal transcripts to the animal. And the quantifications, in this case, I have them shown here. So because of this decrease in the multiciliated cells, we were curious about, what is happening alongside that change. Are there cells there that don't know what to do? Are they confused? Are they dying? Are you not making, are they not surviving? Or perhaps are they not proliferating correctly, the precursors that would make the multiciliated cells? And so to explore that question about what else is happening in these nephrons, Hannah did a number of other studies where she looked at the populations of cells that are monociliated. So if I just go back, she could look at unique markers of these monociliated cells here that she colored in green and ask the question, are those cells there, or are there changes to them as well? And in fact, what she found was that when she knocked down estrogen-related receptor gamma, there were fewer multiciliated cells shown here with in a wild type. We have the normal number where each of these green areas indicate a cell with transcripts encoding this ODF3B, which is specific to multiciliated cells. And there were many fewer of those cells in the deficient animals. Here on the right is a space filling model so that you could get a sense of where those different cells are actually located, that as she analyzed the Z-stacks in her confocal. And what she found when she counted the cells and did that analysis is that there was a change in the identity of cells within the kidney. So there were fewer of the cells that were multiciliated, but cells here that are depicted and assessed by expression of TRPM7, she found an increase in those cells. So many more cells here in red than in a wild type. And another way to represent that is shown up here with this graph, where you can see that there is a ratio of the mono to multiciliated cells, which is altered when we compare the wild type to the knockdown animals. So there are many more cells which are monociliated in identity. So I guess I should call them monociliated transporter cells in the morphans. And other assays that I'm not showing here, Hannah did look to see whether the numbers of cells were affected. Is any aspect of cell dynamics, is that being changed? So are there alterations in proliferation or death of cells, and could not detect any of those kinds of changes. And so because the ultimate identities here are shifted in proportion, what our current hypothesis is that, and we've concluded so far, is that the cells that are precursor cells are having a change in their fate. So fewer cells are going down the path to become a multiciliated cell in the absence of estrogen-related receptor gamma. Okay, and then in addition, they are not making cilia correctly. So at this point, Hannah and I were both curious about whether or not these changes we were seeing in what type of ciliated cell is made and how cilia are made in that cell, if this was all just happening in the kidney when you had this interruption in estrogen-related receptor gamma, or if this was broadly, more broadly applicable. Is this relevant to how ciliated cells are developing in other places in the embryo? And so what Hannah did was start looking at different other places in the embryo, and we were amazed to observe the following, and that is that there were defects in ciliogenesis in every other tissue actually that Hannah has looked at so far. I just have two examples shown here today. So in that otic vesicle, so in that ear where there are cilia in different functional regions of that ear, both and these are termed the crista and the macula. In the case of the wild types, these are ciliated, beautifully ciliated. You can see a zoomed in picture here on the right. In the animals with the knockdown, there was really a dramatic absence of cilia within the otic vesicle. And in addition, at younger stages, so here is that 12-hour-old embryo again with that Kupfer's vesicle that I alluded to at the beginning, which sets up the left-right, helps to set up the left-right body axis. When Hannah looked at wild types compared to her deficient animals, there were fewer cilia in these structures, and they were also shorter within the Kupfer's vesicle, and that length is quantified here just as an example of our quantifications. So Hannah concluded is that this gene is quite important for ciliogenesis across multiple tissues of the body. Okay, so our next question is, or challenges in figuring out, how does this gene fit into the bigger picture of the genetics of how cilia are made, or how cells are making choices in development to form one or more cilia. And what we wanted to do was to try and figure out, where does this gene fit in? And what occurred to us as Hannah was making all these observations is that all the things she was noticing from back from the very beginning, where we saw pericardial edema, all the way to seeing defects in cilia in these other locations, these were all really reminiscent of changes in the levels of signaling molecules known as prostaglandins or prostanoids. And in beautiful previous work, other labs have shown that these signaling molecules are extremely important to make cilia. And I've got the citation here nestled with the cartoon from the authors that first reported this in a really beautiful paper in Nature Cell Biology. And they found this to be true in zebrafish and in mammalian models, and a couple of, several different mammalian models and cell line models, that this was true. And so these molecules that I'll talk about now in the rest of the talk, known as prostaglandins, are molecules that will influence or can be used for cells to signal to one another. And they are produced through biosynthesis within a cell through really a two-step process. It's simplified here. And starting with this precursor, arachidonic acid, there are enzymes which will convert arachidonic acid into these bioactive molecules, prostaglandins, which are lipid-derived signaling molecules. So these lipid-derived molecules can either be transported out of cells, or they can move across membranes without such transport. But when they leave cells, what was found with regard to ciliated cell development is that they were critical for binding to dedicated receptors and for influencing intraflagellar transport. So in the absence of prostaglandins, the process of making cilia was disrupted, and very short cilia were observed in animals that were deficient, for example, in these COX enzymes, or deficient in other studies that we've done when we've looked at deficiency of PGE2 specifically. And so what was ultimately known before is that you needed these enzymes that produce prostaglandins for PGE2, just one prostanoid, to be made to have normal ciliogenesis. And again, embryos with defects in either of these two had the same constellation of phenotypes that we observed in the estrogen-related receptor gamma knockouts. It really begged the question, does our gene that we've been studying relate in some way to this pathway? And so this is the next thing that Hannah tested. And to begin exploring this, the first most obvious place to start was just to look at embryos and assess whether there was any change in the amount of bioavailable prostaglandin E2. And so Hannah performed this test. This is an ELISA that she performed, or the results of an ELISA she performed, and she found that the relative concentration of PGE2 was significantly less in animals deficient in estrogen-related receptor gamma. And then with that finding in hand, she took one step upstream and looked to see what the status was of transcripts transcripts that corresponded to the biosynthetic enzyme that produces that active prostaglandin E2. And through qPCR, what she found was that there was a significant decrease in the number of transcripts of PTGS1, and which is, if I back up, actually, in the zebrafish, COX-1 is known as PTGS1, so those names are interchangeable. Okay, so she saw that there was a decrease in that transcript quantity, and she then looked at the promoter region that is upstream of PTGS1, and in the proximal promoter, she identified binding sites that would be recognized by estrogen-related receptor. And so with this in hand, she then sought to ask the question, well, if PGE2 is decreased in these animals, and if these transcripts are decreased, could she restore ciliogenesis in her animals by restoring either of these two phenotypes? And so here on the right, just showing you example pictures from one region of the kidney, but she's looked throughout. So first we have the wild type kidney with the normal amount and density of cilia that are in the nephrons, the dramatic decrease in animals which are deficient in estrogen-related receptor gamma. In cases where she grew those embryos in the presence of a stable form of PGE2, she was able to restore ciliary number and length, and as well as the number of ciliated basal bodies was restored to wild type levels. And similarly, the provision of RNA that encoded PTGS1, this was also sufficient to rescue her phenotypes in her animals. And so this has led us to the following working model, where we hypothesize that this estrogen-related receptor gamma A is responsible for signaling in such a way that it is controlling the level of expression of this prostanoid biosynthesis enzyme, PTGS1, again, or COX, known as COX-2 in mammals, which is responsible for promoting or responsible for the first step in prostanoid synthesis and in that production of PGE2. And that is this key molecule that's promoting the formation of cilia. Okay, so that is where our model is at present. We have another component that we have actually been testing that we think estrogen-related receptor gamma A synergizes with. And just for the sake of time, I wanted to make sure we had lots of time for questions if folks had them. So I didn't include that here, but we do have one more bit of hypothesis in our working cascade here that that you can see and find in that preprint. But I wanted to make sure I had enough time to acknowledge all the folks who have done this work and who on a daily basis are just fantastic to work with. So again, the lion's share of the work here has all been done on this story by Hannah Wesselman. She's a PhD, a fourth-year PhD candidate in my lab, but we are assisted in our scientific community by all the graduate and undergraduate students that that work with us, and we're exceptionally grateful for funding to support our work on ciliogenesis. And Hannah specifically has received a graduate fellowship, several graduate fellowships over time to support her. One from the Center for Stem Cells and Regenerative Medicine here at Notre Dame, one from the Graduate Women in Science, the GWIS Foundation, and most recently a grant from a precision medicine center here on campus, known as the Warren Center for Drug Discovery. And with that, I'm happy to take any questions.
Thank you so much, Professor Rebecca. Amazing data, very interesting presentation. So we are open to question now. Usually we open to the students' questions first, and then for the professor's question. If the students have any questions, you can open the mic and the camera. Miriam, we have any question in you?
Not right now. I have a question if you don't have any students' questions over there, because we usually ask the students first. We can give them a little bit of time for the data to sink in as well. So
Yes.
Luisa, if you have any questions over there, we can take them. If not, I can start.
You can go, Miriam.
Yeah. Okay, so, well, first of all, thank you very much for all the data you showed and all the beautiful questions you're asking. And I was intrigued by the rescue that you were able to have with the PGE2 and cRNA in the zebrafish. And I was wondering if you treat the wild type fish with the PGE2, do you get an increased cilia production? And if you think that it seemed that when you used the RNA, you got longer cilia, and they were not organized as you saw in the wild type. So I was wondering if that is something that you'd be interested in checking as well, the type of organization that you can get. Thank you.
Yeah, thank you. Those are wonderful questions. So just in case anyone missed those, the question is whether in the zebrafish, when we treat with exogenous PGE2, or if we provide those PDGS1 RNAs, do we have an elevation in ciliated cells or an alteration in the length of the cilia? And we, in a wild type animal, and my lab has not observed that. And several students have done those experiments. We first did them when we studied PGE2, when Amanda Mara in my lab, Dr. Amanda Mara when she did some work on prostaglandins, she did not see an increase in wild types in terms of cilia length or numbers of, say, the multiciliated cells or the numbers of cilia in those bundles or anything like that. There seems to be there is just, I don't know how to put it, except I think of it as a threshold, like we just can't get more. We don't have a gain of function phenotype in a wild type. And so that was true in Amanda's hands. A subsequent student in my lab, Joe Chambers, in the course of doing his studies and doing all the controls, he similarly, we've never seen an increase in any of those cilia features, and Hannah similarly repeated them, and we have not seen that. So I don't know why, but we seem to be at, there is some maximum, you don't get any more cilia, and I don't know why that is, but it's intriguing to me, but I don't have a satisfying answer as to why, but yeah, we've just not seen that. Yeah.
Okay, no, that's interesting, because you'd think that if you'd had an inflammation episode early in life, you'd get all messed up kidney or maybe transport. So it's interesting to have a cap of as much as you can get.
Yeah, there's some safeguard or cap or something. Yeah.
And do you see a direct? Do you see on the promoter of the COX? So I wrote down the zebrafish homolog. The PTGS1, you see the responders for, or at least binding sites for estrogen receptor, or would it be a secondary event to the estrogen signaling?
That's a great question. If there is, if there's a step in between, so our hypothesis is that estrogen-related receptor is ultimately binding and promoting PDGS1, but there's another factor that we have identified that we think might be interacting with estrogen-related receptor that has its own binding sites as well, whether they're themselves interacting and binding, or multiple factors are binding to recruit other transcription factors. We don't know yet. So I think there's cooperation. I think the best way I can answer that is that there's likely cooperation and other transcription factors involved in promoting PTGS1 expression. So we don't think it's a lone lift by estrogen-related receptor 1, but we don't know if we don't know what the order of those events are and who all the players are. So I think those are great areas for future study.
Can I also ask a question regarding PGL2 function?
Yeah.
That I was thinking about the importance of this gene you have shown about the degeneration of ciliated cells, but the production of prostaglandins, they occur into the ciliated cells, or there are other cells in the kidney of zebrafish that are also enhancing this promoter of this gene to help to produce this kind of molecule and helping the generation of ciliated cells.
Are there other cells in the kidney that produce prostaglandin?
Yes, this gene could also be upregulated in other kind of cells that are not the ciliated cells, but regulating the production of prostaglandins.
It's a great question. As far as we and others have observed, all the cells in the nephron will get at least one cilia, but nearby cells are also likely producing prostaglandins. So I suspect it's not just kidney cells signaling to nearby kidney cells. There could be cells in other nearby tissues that are producing locally prostaglandins. As these things, they do move and diffuse. They typically have a very short half-life. So when they are produced and secreted by a cell, they'll often just influence cells nearby because they are, they don't have very long stability. But it's formally possible and probably likely that other nearby tissues are part of the equation. But the nice work by Jin and colleagues has the strongest and really elegant evidence that that kidney cells are signaling to kidney cells, and we've certainly seen expression of our lab has seen expression of several prostaglandin receptors in the kidney cells. But again, prostanoids can get in and out of cells without that receptor. So, but we have seen those receptors on the cells.
Luisa, we have a question from Bruno over here.
Hi. We were actually discussing here, and we're not sure if we missed something. But have you actually defined which cell is making the prostaglandin E2 in your setting? Is actually the MCC that is making the E2?
We have not. That's an awesome question. We have not defined that it's the multiciliated cell precursor. The data that's out there looked at cells in the kidney, but they didn't look at the markers of multiciliated versus transporter. So we have not done that experiment yet.
Right.
Yeah.
One other thing that I was thinking about looking back when the early experiments with the COX-1 and COX-2 knockout in mice is that the COX-1 mouse, the female, had delayed labor the pregnancy would extend, and sometimes fetus would die just because they were not being delivered, and these could be replaced by the COX-1 deficiency could be eliminated, the phenotype with LPS injection, because COX-2 would then make all the PGE2 that was needed in this setting. So I was wondering if is there any role for COX-2, PTGS2 in your data, and also these data has any implication on any potential renal toxicity of the aspirin-like drugs that you may encounter in humans, for example?
Yeah, thank you. Those again are all really great questions. So the first is the role of COX-2 in all of this. So we have done experiments that we have published where there seemed to be a role that COX-2 was playing. We have not done QRT to quantify in the embryo or in kidney cells in particular. So that's just something we haven't gotten to yet. Amanda Mara looked at these genes a little bit. So that it requires further study. We don't know yet. It's a great question. And then your other question is about toxicity of
aspirin-like drugs.
It's not something that I think people have explicitly tested, whether there is an effect on the cilia of cells in a mammalian kidney. Our cilia in the zebrafish are all moving, so they are all motile, whether they are mono or multiciliated. In the case of the healthy human kidney, for example, the cilia are thought to be all mechanosensory, so they are not moving based on their structure. But in different, and I think I skipped over talking about this, but in kidney, in a number of kidney diseases, multiciliated cells have been detected in people in the adult kidney, and so they appear to be showing up with a number of different kidney conditions. There hasn't been a really good exhaustive study of that and about what time in these diseases, but a number of different pathological conditions, multiciliated cells are observed in humans. But people so far have hypothesized that is related to potentially flow in those kidneys, that you have multiciliated cells showing up as a response to try and evacuate fluid and keep fluid moving. Because in our kidneys, we don't need cilia that move. There's high blood pressure, so the fluid's going through. That there's a different situation in the zebrafish. You need cilia to be shepherding the fluid through. So with all that said, depending on health or disease, it might be different. I don't think anyone has, just really probed that. It's a really interesting topic, though. Yeah.
Thank you very much. It was very interesting work. Thank you.
Thank you.
So Bruno, I think we don't have more questions for now here in Zoom.
We actually have one late question from one of our students here, Lucas.
Okay.
Hello, thank you. We were wondering if we understood that in your model, the adult zebrafish ended up dying, but we were wondering if you could revert this scenario with the PGE2 and with the mRNA in adult individuals and not only on embryos. If you could talk a little bit about that.
Yeah, so The adult zebrafish, delivery of messenger RNAs, this would be very difficult. You would have to package in viruses or something like that. We haven't done that kind of attempt in an adult zebrafish. You could, however, expose adult zebrafish to drugs as you would any other adult animal with, an IP injection or something like that. So you could do the experiment to provide a exposure to prostaglandin E2, for example. We have not done that kind of experiment in the adult, either by an injection or baiting adults in the prostanoid. So I'm not sure what effect we would see. I'm sure it would be related, of course, to dose and what dosage would actually get into the animals. These zebrafish embryos are very tiny. They're very easy to do this delivery of these agents to them. And so in the adult, we don't know. It's an interesting question.
Thank you.
Yeah.
Miriam, do you have more questions during you?
No, I think we are all set here. Just thank you.
Okay, so thank you so much, Professor Rebecca, for being here with us today. It was an amazing presentation. Thank you so much.
Thank you so again for having me. It was a pleasure to interact.
Yes, it was a pleasure. And just a message for the audience that in the next week, we'll have with us Professor Martin Bonamino from INCA that will talk about the genetic modification of T lymphocytes for cancer immunotherapy, okay? So I see you guys next week. Thank you again, Professor Rebecca.
Thank you.